Great. Hi, everyone. We're going to start a chapter on immunology, or acquired or adaptive immunology. Um, I just wanted to mention regarding this slide and this picture. This is actually the Russian army who forcibly vaccinated people against smallpox. Um, they used a live vaccine, of course, which is no longer used, right? And it killed between 30 and 50% of the people who were vaccinated with it. So for a long time in Russia, the older generation warned the younger generation against vaccinations, told them not to be vaccinated. Of course, today we use much safer vaccines. They're either attenuated or they are acellular, or in the case of the vaccine that's being developed for COVID-19, it's actually an RNA vaccine, so there's no chance of any type of infection or symptoms. Okay, so we talked in the last chapter about your innate immunity, and these were factors and mechanisms that you are born with and that are immediately available to you. But this is the immunity, your adaptive immunity or acquired immunity, that your immune system mounts only in response to the disease agent. So once you have an exposure, your immune system would mount this immune response against that particular disease agent. So it would be highly specific, but it would actually take a little bit of time to develop. And in the meantime, you would probably get sick. Okay, but like you probably have some experience with, either getting a cold or getting the flu and then having immunity to it. You actually oftentimes, not always, have memory against that particular agent, so that when you encounter it again, you might feel a little bit under the weather, but you probably wouldn't even know that you came into contact with that agent at that point, because you would mount such a rapid memory response, and it would be such a much stronger response that you would be able to fight that agent off when you encounter it for the second time. Okay, so our innate immunity was the focus of the last chapter, chapter 16, and now this acquired or adaptive immunity is our focus for chapter 17. Okay, so it's highly specific, and it's only going to develop once you've had an exposure. That exposure could either be a natural exposure or an artificial exposure. So a natural exposure would be like, say, for instance, if you caught a cold and you mounted an immune response against the cold. An artificial exposure would be if, say, for instance, you got a vaccine. Okay, so that's an artificial exposure. But in both of those cases, your active immunity, your immune system would be creating the response in response to either that natural or artificial exposure. A passive acquired immunity is when you are given ready-made antibodies. And so we've heard a lot in the news recently about COVID-19, the convalescent plasma, right? So that would be passive immunity. So where you're giving, and it would be artificial, where you're giving antibodies from a different person who has already been exposed and fought off the disease to a person who is currently fighting the virus for, and you're giving them those antibodies so that the antibodies can attack the virus and eliminate it, and presumably that person would be able to recover faster. So there is a passive natural transfer of antibodies, and that would be like the antibodies that are passed from a, a, a woman who has just given birth to the child through like the colostrum. Okay, that would be like a natural passive transfer of antibodies. On this next slide, I'm showing some examples of, like, for instance, your naturally acquired active immunity. Okay, that would be like if you caught a cold. Okay, but your body is still fighting it off, so that's your active immunity. Artificially acquired active immunity would be like when you get your flu shot. Okay, so it's artificially acquired because it's not a natural exposure, but it's still your active immunity where you are creating the immune response. A naturally acquired passive immunity would be the transfer of antibodies from mother to child across the placenta or in the colostrum. Artificially acquired would be like what I was talking about with the convalescent plasma from people who have had the coronavirus. Also, anti-venom for rattlesnakes, rattlesnake antivenom. That is another type of artificially acquired passive immunity. For that, you actually take the toxins, and they're inactivated, but you inject them into a different animal. Usually, it's a horse, and the horse creates an immune response against those inactivated toxins. So the horse doesn't respond like it's been bitten by a rattlesnake, but it has the toxins have been inactivated, but there's still proteins that can elicit an immune response, and so the horse would mount an immune response against those toxins. And then plasma, so blood would be drawn from the horse, and the plasma would be separated out, and the plasma would contain the antibodies, and then those antibodies could be used, like, say, for instance, if a person is bitten by a rattlesnake, you could give the person that antivenom, and the antibodies would bind to the toxins before the toxins could bind to your cells and cause a problem. So your natural immunity is going to take a couple of weeks to develop, and this was what I was talking about just a minute ago. So it only occurs in response to an exposure, and it takes a while to develop. And in the meantime, okay, you could get sick, but you're going to create a memory fraction, and that memory fraction could last a while. They're talking about, like, with the coronavirus, that they don't know how long that protective memory lasts. But if you think about, like, some of the vaccines that you got as a child, you're never boosted on those. So the memory actually lasts a lifetime. For others, like the TDAP, you're supposed to get boosted on it every 10 years. So those don't last a lifetime, but they last a pretty long time. But with the coronavirus, they're saying they're not quite sure how long the protective immunity may last. So this is kind of variable. With passive immunity, where you're just getting the antibodies, that is only going to last for as long as the antibodies themselves last. Okay, and antibodies in relation to other proteins are more stable than just your average protein because they have a lot of disulfide linkages. And so we'll see the structure of an antibody in a minute. So in general, they are very stable proteins, but okay, they're only going to last weeks to months. Okay, they're not going to last for a year. They're not going to last like your memory cells to an exposure would last. So they could last weeks, they could last a couple of months, a few months, but after that, for any type of immunity, you would need to rely on some type of active acquired immunity. All right, so I mentioned that the toxins in rattlesnake venom could create an immune response. I just wanted to talk about antigens for a second and what makes a good antigen. Antigens literally, like if you break the word down, mean antibody generating. So these are substances that are going to elicit an immune response. And the best antigens are large proteins. Okay, and the reason why the proteins need to be large is because the antibody is going to recognize a particular fold or a particular shape of the three-dimensional folded proteins. So as a protein gets smaller and smaller, and if you just had like a small peptide, a peptide might not be antigenic because it may not have enough secondary or tertiary structure. A good antigen could be a glycoprotein, okay, so a protein that has a carbohydrate attached. Some polysaccharides are antigenic as well, okay, but the best antigens would be large proteins, okay, and you can see in this figure some examples of antigens, okay, like for instance, the flagellum, okay, the flagellum is made of the individual proteins, globular proteins, flagellin, okay, remember arranged in that helical structure, the filament of the flagellum. Okay, so proteins, all right, that's going to be antigenic. The pilus, okay, or the fimbriae, those are made up of the protein pillin, all right? So again, those would be antigenic as well. Also the cell wall, all right? So the cell wall, the peptidoglycan is made up of peptido, right? Protein, glycan, sugar. Okay, so this is going to be antigenic as well. Okay, so all of these things that would be on the surface of the microbe, the pathogen, that would be exposed, okay, your body could actually elicit an immune response against those proteins. Okay, so say for instance, like in this figure right here, that is the protein flagellin. Okay, that's the protein flagellin. You could actually, on that folded protein, have several sites that could elicit an immune response where you could have antibodies directed against several different sites on that particular protein. And those sites would be called epitopes. okay, or antigenic determinants. Okay, that's where the antibody is going to bind. All right, so the antibody would be raised in response to those particular folds on that large protein. Okay, so you we'll actually talk about the pregnancy tests, and for the pregnancy test, you need to have two different antibodies to that hormone, and for the test to be able to work. So there's an antibody against the alpha determinant and an antibody against the beta determinant. And that's how that test works, is that you need to have two antibodies raised against that particular antigen. Haptens are substances that are so small that by themselves they do not elicit an immune response. Okay, they're not antigenic, but when they are bound to a larger molecule, then they can elicit an immune response, because now they're part of this larger molecule, and the antibody can recognize them in the context of the larger molecule. Okay, because they're just like one part or one shape of that larger molecule. So when we talk about your acquired immunity, we talk about your B cells and your T cells. And your B cells and your T cells are lymphocytes, and they originate in your they're from your stem cells, which originate in your bone marrow. So your B lymphocytes are going to continue to develop and differentiate into mature B cells in your bone marrow, but your T cells are actually going to travel from your bone marrow, okay, via the blood to your thymus, okay, where they are going to develop and differentiate into mature T cells. Okay, so we'll see that your B cells and your T cells are going to carry out your immune response. And your immune response, your active immune or adaptive immune response, has a dual nature. So it can actually try to eliminate the pathogen before it infects cells, but once it infects cells, it can carry out a kind of a second type of attack, which would be to attack the infected cells by a cell-mediated response. So the first part of your immunity, which is carried out by antibodies, so the antibodies binding to viruses or the antibodies binding to bacteria, okay, kind of marking them for elimination, that's carried out by your B cells, and it's your B cells that create antibodies, okay, and this is what's referred to as your humoral immunity. Now, if that fails to ward off the pathogen, and the virus actually infects the cells, or the bacteria infects the cells, then, okay, your T cells can carry out this cell-mediated response where they target the infected cells and kill them. Okay, so we'll see how your antibodies can opsonize, right? We talked about that with your innate immunity. They can mark a pathogen so that it is more easily recognized by phagocytes. And they are going to be most effective against pathogens when they are outside of cells. Once your cells are infected with a virus, remember how we talked about how, like, especially enveloped viruses would be expressing their glycoproteins on the surface of the infected cell, because when they bud from the surface of that cell, they would need to have their viral glycoprotein spikes in the envelope, in their envelope. So when your T cells can recognize these antigens that are that okay, like the viral glycoprotein spikes that are embedded in cell membranes, and carry out an immune response to attack those cells and kill them. Okay, so we'll talk about your cell-mediated immunity as well. It's important also to note, and we'll see this when we talk about T-dependent versus T-independent antigen, that your T cells also play a role in stimulating your B cells to produce antibody. So normally we associate B cells with your humoral immunity and T cells with your cell-mediated immunity. But your T cells, okay, specifically your T helper cells, also play a role in your humoral immunity. Okay, so as we talked about how your T cells develop in your thymus and your B cells develop in your bone marrow, and I talked about them differentiating. Okay, so they start out as stem cells, but then they differentiate into these highly specific cells that that can recognize basically, any antigen out there. So they express antigen receptors, and so each B cell would express around 100,000 antigen receptors, and they would all be identical. But then between the different T cells, okay, every T cell is expressing an antigen receptor that is different, okay? And you have this vast array of B cells and T cells that are expressing all of these different antigen receptors that can recognize virtually any antigen out there, and that's how you're protected, okay, because you can mount an immune response against these pathogens once you encounter them. So you've got like all of these B cells and T cells, but based on your personal natural or artificial exposure, you might only activate a small fraction of those, okay? And everyone's immunity is different based on, where they've traveled and what exposures they've had. Okay, so you've got this vast array, and you probably won't use most of them, okay, but they're available to you, okay, and the antigen receptors, okay, are proteins, all right, and they have different shapes, okay, to bind to the particular shape of the antigen. And once they bind the antigen, okay, you're going to mount this immune response against it, okay, and that's what's referred to as clonal selection, and we'll go through that in just a minute. Okay, but your T cells and B cells, okay, they develop in the thymus and in the bone marrow, respectively. And as they develop, okay, they are expressing these antigen receptors. And once those antigen receptors are expressed, okay, on the surface of the cell, okay, then you have a differentiated B cell or T cell, and they can go out into your blood and your lymph and bind to antigen and mount an immune response against it. I mentioned it on this slide right here, 2,000 antibodies per second. Okay, and so they're very short-lived. All right, they only can make this high output of antibody for a short amount of time. Primary and secondary response is a round of clonal expansion like this, or clonal selection. So this would be the primary response right here. Okay, what we're seeing right here in this figure. And then, okay, for the secondary response, if a memory B cell comes into contact with the antigen again, you would go through a whole second round of clonal expansion, where you would make more of the plasma effector cells and you would make more memory cells. Okay, and you probably know from having vaccines that sometimes a second vaccine is necessary, and even a third vaccine is necessary to make a strong response, right? So in the beginning, if you just have the primary vaccine and you don't have the secondary vaccine, you might not make a response that has a good amount of memory cells to give you a very strong secondary response. So we'll talk about this with respect to the types of the classes of antibodies that are produced, and the time it takes for those antibodies to be produced, and then how long that response lasts. And of course, it kind of depends on the antigen, how long the response lasts. At the beginning of this lecture, I said that some of the vaccines that you get as a child, you're never boosted on, right? And then others you need to be boosted on every 10 years or so. Okay, so with the primary response, antibodies would be detected 4 to 7 days after contact with the antigen. And this will be initially, the first antibody to be produced would be IgM. And then IgG, there's class switching where then you would have IgG production, and the amount of antibody is going to come to a peak, or its highest concentration or titer, at about 10 to 17 days. Okay, so approximately 2 weeks after you come into contact with the antigen. Okay, so for that primary response, you are most likely going to get sick, right? Okay, but like we saw with the clonal expansion, you make a number of effector cells, and then you also make, a fraction of memory cells. And if you ever come into contact with that particular antigen again, those memory cells, would be activated and initiate a second round of clonal selection. Okay, and so this is going to occur much faster. Okay, so instead of having the amount of antibody, peak at 14 days approximately, the amount of antibody could peak at 2 days. Okay, and it's going to be a higher peak. Okay, there's going to be more antibody. It's going to last longer. All right, we'll see that on a graph, and it's going to be mostly IgG. All right, so here's the graph right here of the primary response and then the secondary response. So you can see, just like if you looked at both graphs just quickly, that there's a bigger lag period with the primary response than with the secondary response. Also, the total antibody for the secondary response is a lot higher, right? Okay, so you make more antibody. It's a stronger response. And then for the primary response, you can see where the total antibody plateaus and then it declines. Okay, and it basically goes all the way back to almost like zero, right? But look at for the secondary response, okay, that doesn't decline back to zero. That is like a tail, all right? So that's what I mean when I say it lasts longer. And it's mostly IgG. Okay, so you can see that there's a big part of that curve is IgG. Okay, so you start with IgM and then you make IgG. All right, and so below that you can see clonal expansion. All right, so that fraction of B cells that has a complementary antigen receptor would bind to the antigen, okay, and then it would be activated and it would divide and differentiate into the two populations, which would be the memory cells and the plasma effector cells. Okay, initially the plasma effector cells are making IgM, and then they make, okay, for so there's a slow rise in IgM, and then there later is a larger peak of IgG. So you can see where it switches classes of antibodies, and then you have your memory fraction, and it's the memory fraction that would be activated in the second exposure, okay, and that is going to make a much stronger response, okay, and in a shorter amount of time, and it's going to last longer. All right, so when you have this secondary response, you might feel a little under the weather, but you may not even ever know that you were exposed to anything or feel sick. So if your primary response is, say, a vaccination, and you know, it's not like it's a live vaccination, right, like the smallpox, it's, you know, a killed or acellular toxoid vaccination, something like that that couldn't make you sick, and then you come into contact with the disease agent, and in the secondary response, you just make antibodies and you don't even, you know, realize that you ever came into contact with something. I mean, this is true protection, right? Where you're never experiencing this particular infectious disease. Okay. I just wanted to talk about some different antigen types. When we looked at that first graph of clonal expansion, there were T cells in there. I didn't mention them, but I think at the beginning I mentioned how T helper cells can play a role in your humoral immunity, right? Okay, and so we'll describe more on how that happens. So most antigens are T-dependent antigens. Okay, so, you know, we're thinking about the antigen here, right? And we said that glycoproteins are good antigens, proteins are good antigens. They need to be big. They need to have like the secondary structure, right? Okay, so most antigens are T-dependent antigens, but there are some antigens that are T-independent, okay, which means that the T helper cell is not going to play a role here. And these antigens are usually like, they could be polysaccharides, where they have repeating subunits and they might make contact with multiple receptors on the surface of the B cell. And in this case, they do not require the T helper cells. They can be activated, but it is a very different response where no memory cells are made. Okay, you can see in the figure, right? And you're only making plasma effector cells, which make the IgM. Okay, you don't make the IgG. So with this type of antigen, there would be no memory. Okay, so that's important. So the T-dependent antigen is more typical, and whereas B cells can bind to antigen directly, T cells have to have antigen presented to them. Okay, so the B cell actually becomes the antigen presenting cell, and it will internalize that antigen and process it into peptides and then present it on its surface in the context of these MHC molecules. Okay, in this case, it would be an MHC class 2 molecule. So MHC stands for major histocompatibility proteins. These are glycoproteins, and the two classes that we are concerned with are 1 and 2. 2 are present on the surface of antigen presenting cells. Okay, like this B cell right now is an antigen presenting cell. Macrophages are also antigen presenting cells. Okay, macrophages that can phagocytose pathogens and process them. Okay, they could present that on their surface and present that, okay, to a T cell. So the B cell becomes an antigen presenting cell and presents this antigen on its surface. And this is where the T helper cell comes in and binds to, okay, that antigen in the context of that MHC class 2 molecule, becomes activated, produces cytokines, okay, which activates the B cell to grow and divide and differentiate into the two populations, okay, the memory population and the effector population, okay, which is the population that is making the antibodies. Okay, so here are some functions of antibodies. Antibodies are super important. So we talked about how IgM with its 10 binding sites, right? So it's five antibodies bound together by the J chain. It has like 10 binding sites. Each of the antibodies has 2 binding sites. That it is especially good at clumping together or agglutinating pathogens. Okay, so it clumps together all of these pathogens, and then something like that can be more easily disposed of or phagocytosed. Neutralization. All right, we'll talk more about this when we get to chapter 18 as well. Neutralization, if you have an antibody that can bind to a toxin, okay, or a virus, okay, it can prevent the virus from infecting the cells, okay, or the toxin from having its particular deleterious effect. IgG is actually a very good neutralization antibody. Also, okay, from our discussion of innate immunity in chapter 16, we also saw how antibodies can initiate the classical pathway of complement, right? Okay, which can result in the lysis of a pathogen through the MAC complex. Okay, it can also stimulate inflammation and phagocytosis. Okay, this is just a little summary slide right there for you. Okay, so I just mentioned that T cells do not bind to antigen directly like B cells do. So they have to have antigen presented to them. Okay, and so we just saw how the B cell actually internalized that antigen and processed it into smaller pieces, and then presented it on its surface to the T cell. T cells are not going to make antibodies, okay? They're not part of your humoral immunity. They have other functions, okay? So we saw how the T helper cells could secrete cytokines, which could activate the B cells to make antibody. We're also going to see how T cytotoxic cells can be activated to become cytotoxic T lymphocytes and attack virus-infected cells or even cancer cells, okay, tumor cells. Okay, so the antigen-presenting cell, okay, like the B cell would have the MHC class 2 glycoprotein on its surface, okay, and the cell-mediated response, okay, so the cytotoxic T lymphocytes, that is usually going to start with antigen being processed by macrophages, okay, and then that processed antigen would be on the surface of the cell, okay, of the macrophage with the MHC class 2 molecule, and then that would okay, so these are the two classes of T cells that we are going to be most interested in, and those are the T helper cells, okay, and then also the T cytotoxic cells. Okay, the T cytotoxic cells carry out a function that is a lot like the natural killer cells of our innate immunity. So they kill their targets by like punching a hole in them and causing cytolysis. Okay, we'll talk more about that in just a minute. Now, it is a thing for your immune system to attack a self cell, right? Okay, so it knows that it is a self cell, okay, but it is infected with a virus, okay, and so that is the function of the MHC molecules. Okay, the MHC molecules are on antigen-presenting cells. They're also on nucleated cells. Okay, the MHC class 1 molecule is on a nucleated cell. Okay, but when antigen is presented in the context of that MHC class 1 molecule, then, all right, it's okay, basically, for the cytotoxic T lymphocyte to kill that particular cell. Okay, and it is also important to note that the T cytotoxic cells, okay, which become the cytotoxic T lymphocytes, can also recognize self cells that have been altered, such as tumor cells. Okay, so tumor cells would be expressing a protein, okay, on its surface at an inappropriate time in development, and your cytotoxic T lymphocytes would recognize that and could target those cells. Okay, so there are two classes of glycoproteins which are referred to as clusters of differentiation, or CD. So CD4 and CD8 cells. You may have heard of that with respect to HIV. Okay, HIV infects CD4 positive lymphocytes, which would be your T helper cells. So your T helper cells have CD4 molecules on their surface, and those are molecules that bind to the MHC class 2 molecule. And then your cytotoxic cells have the CD8 molecule on their surface, and that molecule binds to MHC class 1 molecules, okay, which are on the surface of all nucleated cells. Okay, so here we're showing, okay, the macrophage, right, which is an antigen presenting cell. So it is going to phagocytose the antigen and process it, present it on its surface, okay, in the context of an MHC class 2 molecule, okay, which would be the molecule that would be on all of the antigen presenting cells. The T helper cell, okay, would bind to, would have its antigen receptor, which recognizes the antigen, okay, and then its CD4 molecule, okay, which would bind to the MHC class 2 molecule, okay, and that would, so it would bind to the macrophage, okay, and when its receptor is bound, okay, it would become activated and produce cytokine, okay, specifically IL-2, okay, and then that activation, okay, would lead to its cell division and differentiation into at least 2 populations. Okay, your book also talks about T helper 17 cells, but the T helper 1 cell can activate macrophages actually to present antigen and also to phagocytose more antigen, okay? And the T helper 2 cells are activating B cells, right, in the T-dependent antigen scenario where they would be playing a role in your humoral immunity. Okay, so the T helper cell, all right, that binds to the activated macrophage, okay, the macrophage that has phagocytosed the antigen and processed it and presented it, the T helper cell that binds to that would produce IL-2, and the T cytotoxic cells would be transformed into cytotoxic T lymphocytes, okay, which can now kill the self cells, okay, which are either infected with antigen, infected with pathogens, or producing an antigen, okay, that's inappropriate for that point of development. All right, so they would be having on their surfaces, okay, for the virally infected cell, they would be expressing viral glycoproteins on their surface, or if for the tumor cell, they would be expressing maybe some receptor on its surface, which would be inappropriate. Okay, so these surface antigens would be referred to as endogenous antigen, okay, and the cytotoxic T lymphocyte is going to recognize that antigen, okay, presented in the context of the MHC class 1 molecule, and target that cell. Okay, and what it does when it targets the cell is it basically shoots this protein perforin at the target cell. Okay, and perforin would create a pore, which, okay, could cause cytolysis, right? and then it's going to pump that cell full of these granzymes, which are proteases, and the granzymes would initiate the process of apoptosis, okay, or programmed cell death, okay, which is a whole process where the genomic DNA of the cell would be fragmented and the cell would go through this blebbing, okay, and there's some good pictures in your book of that, and then the cell would die. All right, so here we have the cell, all right, the pre-cytotoxic T lymphocyte being activated into a cytotoxic T lymphocyte and going off and targeting abnormal, okay, that would be like a tumor cell or virus-infected cells by pumping these, first causing a hole to perforate the surface of the cell, and then pumping the cell full of proteases, okay, granzymes, which would cause or initiate apoptosis. Okay, just one final word on natural killer cells versus cytotoxic T lymphocytes, okay, how killer cells kill. Both are lymphocytes, but remember that the natural killer cells are part of your innate immunity. So we just talked about how cytotoxic T lymphocytes can kill virus-infected cells or could kill cells with endogenous antigen, okay, like tumor cells. They would bind first to antigen presented by macrophages, and then they would attack their targets. So they are highly specific. On the other hand, your natural killer cells, okay, they are going to, okay, in a non-specific way, act on cells that lack the MHC class 1 molecule. All right. So now this is a little bit confusing because we said that the MHC class 1 molecule was on all nucleated cells. Okay, so how do virus-infected cells or tumor cells lack the MHC class 1 molecule? Well, okay, early on in the infection of a virus, the infected cell may not be expressing the MHC class 1 molecule on its surface. So that in that case, okay, it would be more the target of a natural killer cell than it would be of your T cells. And then also, as far as tumor cells are concerned, oftentimes when tumor cells are rapidly growing and dividing, they may not express the MHC class 1 molecule on their surface either, okay, and so that would make them the target of these natural killer cells.